The human body has around 30,000 genes. These genes code for over 1 million different proteins. How on earth is that possible, though? In school, I was taught that protein synthesis was kind of like cooking. The gene was the recipe, and the protein was the final dish. One recipe equals one dish. But obviously it must not be the case. Protein synthesis starts when a region of your DNA, called a gene, unzips, and an enzyme called RNA polymerase adds RNA nucleotides, molecules similar to what DNA is made of, and forms bonds between them, making a long molecule called pre-mRNA. And this is where part of the magic happens. Pre-mRNA has a lot of misleading parts that don't really code for the protein. These misleading parts are called introns, while the parts that actually code for the protein are called exons. For the mRNA to be useful in making proteins, the introns need to be removed. And luckily, our cells can form just the perfect complex for the task using molecules called SNRPs, which are a combination of small nuclear RNA and proteins. The resulting complex is called a spliceosome. It splices introns out and joins the exons together. But sometimes, some exons are also spliced out to make a completely different final mRNA, and therefore a completely different final protein. This is known as alternative splicing. This means that one gene forms so many different mRNA strands. Then, this pre-mRNA strand undergoes further processing in the nucleus and upgrades, becoming a mature mRNA strand that can safely leave the nucleus into the great unknown of the cytoplasm. Once it's finally out, translation occurs. Not this kind of translation, but instead the kind where the mature mRNA binds to a ribosome and is translated into a specific polypeptide chain, according to the instructions that the mature mRNA is carrying. And now the second part of the magic happens. We have a polypeptide chain that's straight and currently cannot serve any function in the cell. To fix that, the chain undergoes post-translational modifications, or PTM for short. PTM is an umbrella for all the processes that occur after translation is over, hence the name. It includes protein folding into very cool shapes, phosphorylation, the addition of phosphate groups to proteins, glycosylation, the addition of simple monosaccharides and highly complex branched polysaccharides to proteins, ubiquitination, addition of ubiquitin to proteins, methylation, addition of methyl groups to proteins, lipidation, addition of fatty acids, isoprenoids, and cholesterol to proteins. This grows the variety of potential proteins even more. This growth is exponential. And this is how the body manages to have such great diversity in proteins from relatively very few genes. Thank you for watching.